This unit is talking about one of the biochemical tests that we use to help identify unknown bacterium. Yes, you use the microscope to do the gram stain, find the cell morphology, cell arrangement, cell size, things like that. But you are going to have to follow that up with biochemical tests to positively identify the bacteria that you have. So carbohydrate fermentation is just one of those many tests that you can do. It's actually one of the major tests for biochemical tests that will help you figure out what organism do you have. Different bacteria can utilize different carbohydrates, which are sugars, and how they use the sugars is also different as well. So that's why this is a very important test to do. The carbohydrate solution is made up as a broth. It's put in a test tube. It contains the phenol red as a pH indicator. So when it is at neutral pH of 7 or an alkaline pH, it's going to be red. If there is acid present that drops the pH below 7, then it is going to turn yellow. So visually it's very easy to see if acid has been produced as one of the byproducts of breaking down the carbohydrate. There is also what we call a Durham tube that's placed in the test tube. As you put it in as you're making the media, and then you autoclave it. This Durham tube is a smaller, skinnier tube that fits easily inside the larger tube, and it's upside down. It's inverted. So when it goes to the autoclave, it fills up with the carbohydrate broth, whether it's glucose or lactose, whichever sugar you're using. The broth will fill up this tube. So uninoculated, the Durham tube is filled with liquid. It is there to capture gas in case gas is also one of the products from the carbohydrate fermentation. So you're going to be looking for was acid produced and also was gas produced as products of the fermentation process. And as I said, different organisms use different sugars in different ways. And so you will inoculate the tube, and then you will incubate it, and then your results, there's 3 possible results that you could have. If it is red, it is negative. Carbohydrate fermentation did not occur. If it is only yellow, but no gas in that Durham tube, you record it as acid. Typically, we record this as A, and we always use a capital letter. It's just traditional. That's the way we do it. So if you see just A, that means that only acid was produced. If you have the tube and it's yellow, plus there is a gas bubble in that Durham tube, then that means, yes, acid was produced as well as gas. So you would record that as AG, once again using capital letters. So those are your 3 possible types of results from the fermentation of carbohydrates. And so this is some examples. This tube, as you can see, it's written on here, is lactose. This would be negative. It's still red. This is maltose. As you can see, it is yellow for acid. This tube over here was the glucose. It was acid, and look, there's a huge gas bubble in that Durham tube, so this would definitely be acid and gas. You should not ever see gas only, but no acid. So if you see gas, there's always going to be acid along with it. So in our lab, we typically will test for lactose, maltose, glucose, and sucrose fermentation. And like I said, different bacteria will use these differently. So it's going to help you with the identification process.